This lecture will cover the other gram-negative bacteria in the lab tests that have not been previously covered in other lectures. With your Neisseria and your Moraxella species, these organisms, if they are suspected, may be inoculated on the sheep blood agar as well as chocolate agars. And these cultures will be incubated 37 degrees with 3 to 5% carbon dioxide environment, such as regular atmospheric environment. And then to actually culture this organism, you would need to do it right away just to make sure you maintain the viability of it. Now, for culture with the gonorrhea, Neisseria gonorrhea, these organisms should be collected on a specific swab, so it would be a Dacron or a rayon fiber swab. And I will tell you, if you send the wrong swab to the lab, they may reject it. So you need to make sure that you are collecting the right swab for the source, and because if not, then it can actually be an environment for that organism in which it cannot grow properly. So, or it doesn't maintain that organism in transport. So, making sure that you have the proper culture swab is important. And so these organisms for the Neisseria gonorrhea should be cultured on the chocolate agar, but it also can be placed on some of these other agars, which would be like the Thayer Martin or the modified Thayer Martin. And the reason why these other agars would be used, because you can see an image here where you have a chocolate medium. So in particular, if you were doing a vaginal swab looking for to try to diagnose a patient with gonorrhea, then the normal flora that would reside within the vaginal area would be abundant. So if you were to plate it on culture it onto a chocolate agar, then you would get a lot of growth for the other organisms that would be present. And so if you use something that's specific for like a vaginal culture or a genital culture, where some of the antibiotics in there would prevent growth of some of that normal flora, then it helps cut down the organisms that you're looking at. So you can see in comparison here, where at the bottom you have, they actually did this with a rectal swab, where you have a lot of normal flora that has grown on that chocolate agar, versus the modified Thayer Martin media, where just the Neisseria has grown. So some of the different test methods for Neisseria moraxella would include nucleic acid-based testing. And so with these tests, the instrumentation nowadays with the automation will actually look for multiple organisms off of one swab, one sample. And so if you were doing like a GC probe, the gonorrhea and chlamydia probe, then you're looking for both of those organisms off that one sample. Your identification of your organism and how you work it up is going to depend a lot on the source. So it's very important that the sources are labeled properly and that it includes where the source site is. So for example, a rectal source or a vaginal source. Any isolate from a child or anyone suspected of sexual abuse, it definitely has to be identified. It definitely has to be worked up. Isolates from sterile body fluids are completely identified. Isolates from genital sites can have a presumptive identification just based off of the spot test. It's oxidase positive gram-negative diplococci that grew on the select media, so we can presume it to be the gonorrhea. Eye and ear cultures can yield oxidase positive, gram-negative diplococci, and hydrolyzed Trubitin would be considered a presumptive identification for the Moraxella catarrhalis. The carbohydrate utilization test is used to help identify mostly your Neisseria species. And what this test does is uses the different carbohydrates, glucose or dextrose, as it's sometimes referred to, maltose, lactose, and sucrose. And these sugars will all have, or carbohydrates will all have, a phenol red pH indicator in it. So if it is, the organism is able to utilize it, then it will change the pH, and then you would get that yellow color would be demonstrated within that particular tube. So I think in one of the previous lectures we talked about testing these carbohydrates on a tube test. And so here you've got an example of a little bit more of a modified test where you have each of these little whales that have a one of these carbohydrates in it. So you can see here C is control, and so you've got each of these little whales. And then here you have your glucose. It turned yellow, so this would be considered positive. Your maltose is yellow, positive. Lactose is positive, and then your sucrose is negative. You also have a DNase test as well as the beta-lactamase test. But for this particular organism, because it utilizes all the way through the lactose, then it would be the Neisseria lactomycia. So with this, you would also want to avoid putting these into a CO2 environment, because that can affect how these organisms utilize these carbohydrates. So here we have a chart to help compare these reactions in these organisms. There are several species of Neisseria that can also be identified using this test method, but we've not covered those, and they're not always clinically relevant, so we're just covering the ones that you'll probably experience more in your career. And so an easy way to remember these is your glucose is gonorrhea, your maltose is meningitis, and then your Moraxella does not use any of these. So you can see just glucose for gonorrhea, glucose and maltose for meningitis, and then your catarrhalis. Cats don't like sugar. I don't know, do they? So cats are negative all the way through. With your X and V test, there are a couple of different methods. This particular method here requires the use of a nutrient type agar that does not have any sort of X or V in it, so there's no blood in it. And what you do is you actually do one of those lawn sweeps where you culture the organism over the entire plate, and then you place the strips down. And so each of those strips, the little pointed end, has one of the factors in it. So this top one has V, X, and XV. So this particular organism has growth around XV. And so this is considered a positive growth, or it requires X and V in order to grow. Then here in image B, we have V here to the side, X, and then XV here up top. And this organism grows around V and XV, so it requires V to grow but no X. And then here at the bottom, well, you got growth all over this entire plate. So this is considered a negative because it did not require X or V to grow. It grew whether it was there or not. But this is what you're looking for. You're looking for the growth in the presence of X and V, or X and or V. So with our Haemophilus species, we are doing that X and V test. And just a reminder that you can have some organisms that will demonstrate a positive satelliting on the blood agar when they have staph present. And so it could just be that the organism would grow if you have just a staph from a culture. So you've got this that's grown and then it kind of zigzags out and you start getting staph colonies throughout, and then you would have your Haemophilus would actually kind of start pinpointing around that actual colony. Or you could actually do the satellite test where you would streak your Haemophilus or suspected Haemophilus across the entire agar plate, hoping for it to grow, and then you would come back with a streak of, get my colors changed here, you would come back with a streak of Staph aureus down the center, and that would allow that to produce that V. And then what you are looking for is that satellite where that yellow is a little hard to see, but you're looking for the growth of the Haemophilus just around it. Now if it was to grow out here, then that would be negative. It would only grow in the presence of that staph streak. Haemophilus influenza will grow with X and V, so it does require both. So that would be like your satellite, positive satellite test. And then your Haemophilus dicrea would be, it requires just X, so you would see it growing around X and V and X. Campylobacter jejuni would need to be cultured on selective media, so your Campy media. You can have Campy blood agar, or you can have other Campy medias. And so if you were doing a stool culture, the technician is going to set up a couple of different selective agars, and that's because they're going to culture one at 42 degrees Celsius and one at 37 degrees Celsius, just to make sure they differentiate it from another organism that could grow and appear to be similar, that wouldn't be considered pathogenic. With your blood agar, it will grow on a blood agar plate, but it can take a little bit of time to grow. Legionella pneumophila is biochemically inert, so these organisms do not exhibit any sort of biochemical reactivity if you were to test them with any of the previous methods we've discussed about the testing done within the lab. These specimens should be cultured on a couple of different agars if you are suspecting this particular infection, and or if you grew a culture and you suspected that this is what it is, or the gram stain demonstrated this particular organism, then you would do the BCYE agar, which contains the cysteine, as well as one that does not. The pneumophilia, the Legionella pneumophilia, will grow on the BYCE agar with the L-cysteine, whereas your other stuff, it requires this in order to grow. If it does not grow on the BCYE without the L-cysteine, then you would know that it was not Legionella. So these cultures are incubated 35 to 37 degree with humidity for up to 14 days, because it can take several days for these to grow. And then what you're looking for is those gray, white to blue-green glistening convex circular colonies. You can see that image up the top, where you're getting that little bit of a cut glass type look with that color pigment within those colonies.